Hello, everyone, and welcome to today's broadcast, Two-Dimensional and Three-Dimensional Cell Culture. Does dimension really matter? It is presented by Carlemi, PhD, a postdoctoral research fellow at Phamacen Northwest University, South Africa. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Calitz. I will now turn the presentation over to her.
Thank you for that wonderful introduction, Judy. Today, I hope to answer a few questions regarding 2- and 3-dimensional cell culturing and why the right model is of utmost importance when answering complex research questions. In a world where heart disease, tuberculosis, HIV/AIDS, and cancer are among the top 10 causes of death, we as researchers are faced with complex research questions. It is our mission to try and develop the best known It is our mission to try and understand underlying disease mechanisms and attempt to develop new promising drug treatments. To help us in the search, we look to in vitro models to better understand these diseases. The type of in vitro model implemented will determine our successes, and this is where cellular dimension comes into play. Let us look at the human body. The human body is a complex 3-dimensional system with organ systems communicating with each other by biochemical and mechanical signaling. There is also intracellular communication between the various cell types within each specific organ, and you also have intracellular communication within individual cells. Two-dimensional cell culturing has been around for decades, with the first attempt at traditional cell culturing in 1885. It has been an essential tool in the elucidation of many disease states. The culturing conditions are highly controlled with a high degree of homogeneity and reproducibility, allowing for the discovery of molecular mechanisms. This system has also contributed significantly to our knowledge of tumor biology, while stimulating cancer research in terms of drug discovery and development. And it's a simple and cost-effective method. Unfortunately, this system also has its limitations. Keep in mind that this is an isolated, homogeneous population of cell, far removed from the true tumor microenvironment. The system lacks cell-to-cell communication, cell matrix interactions, and most importantly, tissue-specific architecture. Furthermore, cells grown in 2D undergo phenotypic and genotypic adaptations to in vitro conditions. They are also accumulating mutations and is limited in terms of cytotoxicity assays. Your working window for experiments is usually between 2 to 7 days, limiting you to only acute studies in terms of toxicity. On the other hand, animal models have been used for many years in research and drug discovery and remain a prerequisite in preclinical drug screening, according to the FDA. Cancer mouse models have revolutionized our ability to study gene expression and protein function and more closely resemble the human physiology. However, these models tend to be expensive and time-consuming. Furthermore, animal models too frequently fail to reflect the true human tumor biology. These models are in some instances immunocompromised and presents with species and sex differences. Also, not all cancer types have a suitable animal model yet, and most importantly, they come with ethical and moral considerations. With the establishment of three-dimensional cell culture systems, it was found that three-dimensional cell culturing offers a better correlation to this in vivo situation. In three-dimensional cell cultures, cells find themselves in a more natural, geometrical, and morphological shape, resulting in some instances in the formation of in vivo-like tissue structures that provide more clinically relevant data that is more representative of the in vivo condition and comparable to animal studies. Due to the unnatural two-dimensional microenvironment of cells in 2D, important metabolism and biochemical features are altered that has an influence on pharmacological drug responses. These geometric and morphologic differences influences the cytoskeleton, gene, and protein expression that are responsible for cellular responses. One of the most striking differences between 2D and 3D culturing is the culturing time. 2D cultures have a limited working window, usually days to weeks, while some 3D systems have working windows from weeks to months. This allows not only for acute short-term studies, but also long-term chronic studies. Various three-dimensional culture systems are available, and it should be stressed that no one method is superior to the other, and that your research question and available resources will determine which system you will employ. Three-dimensional systems can broadly be classified into static and dynamic or irrigated systems. Static systems, as the classification suggests, is static non-moving systems. These include, but are not limited to, hanging drop cultures, forced floating devices, micromolding and printing devices, galactosylated substrates, 3D scaffolds, and Primaria dishes, while dynamic or irrigated systems include spinner flasks, microfluidic devices, microgravity, and rotating bioreactors. Here at the Northwest University, our research focuses on the multicellular spheroids grown in rotating bioreactors. A spheroid is defined as a spherical, symmetric aggregate of cells analogous to tissue. Spheroids are seen as heterogeneous aggregates with cells that are proliferating or quiescent and retain their 3D architecture with the capability to re-establish their tissue-specific functions. These multicellular spheroids are capable of mimicking in vivo processes such as embryogenesis, morphogenesis, and organogenesis, and possesses several in vivo-like features of tumors, including cell-to-cell interaction, hypoxia, drug penetration responses and resistance, as well as the production and/or deposition of extracellular matrix. In our laboratory, we employ the cell vivo BUMS system that is a clinostat-based system using rotating bioreactors to grow our spheroids. A clinostat is a rotating device that has been around since the 1920s, originally designed to allow plants to experience equal amounts of gravitational pull over 360 degrees. In this way, there is little to no gravitational pull on the plant, allowing the plant to overcome instances of positive phototropism and geotropism. The same principles account for cells grown in a clinostat-based bioreactor, in that the gravitational pull is lessened on the cell. The cell vivo BUM system is a next-generation clinostat-based system that comes with a driving unit, environmental control, and a wireless control system with one of the most user-friendly interfaces. Spheroids or organoids for use for this system can be created by various means. In our laboratories, we prefer to work with agar-well plates from stem cell technologies and sodium alginate. The rotating bioreactors developed by Vivo are fixed volume bioreactors that come already assembled. These bioreactors have a cell chamber with two openings that allow for sampling and medium exchange, and a water chamber with a wick to generate humidity. The cell chamber and water chamber is separated by a 0.2 micron membrane, and the gas exchange labyrinth on the water chamber allows for optimum oxygen and carbon dioxide exchange. Because this is a closed system, instances of contamination and infections within the cultures are kept to a minimum. So to produce spheroids in our labs, we prepare a cell suspension from cells grown in 2D, and then we seed the cells into agar-well plates. The agar-well plates allows us to generate up to 1,200 size-controlled spheroids per well. We then allow the cell suspension to form spheroids within the agar-wells for 24 hours. After 24 hours, the spheroids are removed from the agar-wells and placed in a petri dish. The spheroids are then inspected microscopically to determine their quality, compactness, and roundness. After that, spheroids are transferred to the rotating bioreactors, which is then placed on the bum rotating system. Spheroids are maintained by daily speed adjustments and media replenishment. Spheroids produced in this way can have physiological functionality for a period of 42 days, with spheroids reaching a metabolic equilibrium around 21 days. This means that your experimental design is now not limited to 5 or 7 days as with two-dimensional cultures, but can now be conducted over a 21-day period. We make use of various three-dimensional models produced from a variety of cell lines in our lab. We mostly focus on cancer research, hepatotoxicity, and drug biotransformation. One of our most well-characterized spheroid models is our HepG2C3A spheroid model that we employ in hepatotoxicity studies. Most of the work on this spheroid model was done by our collaborators Dr. Krzysztof Brzezinski and Professor Stephen Fay from Vivo. One of the most fascinating insights into the HepG2C3A spheroids came from the spheroids sectioned and imaged with an electron microscope. The image on your screen was taken of a 21-day-old HepG2C3A spheroid. Electron microscopy revealed microscopic structures within the spheroid similar to that of in vivo liver tissues, namely biochenically, sinusoid channels with microvilli, and even glycogen granules. Work done on the HepG2C3A spheroid grown in microgravity rotating bioreactors found that these spheroids remain physiologically stable for a period of 42 days. This provides us with a metabolically competent homeostatic cell model that permits us to determine drug toxicity and mode of drug action, the evaluation of biomarkers, as well as the study of system biology that requires metabolic functions to be stable for a long time period. These spheroids were also found to undergo significant changes within every aspect of their cellular metabolism, oxidation, transport, growth, and morphology that serves as the foundation of architectural, functional, and physiological differences within cells. Other than the 3D cell culture development, our lab also focuses on the evaluation of traditional herbal medicine for use as potential lead compounds and also to determine safety in terms of hepatotoxicity. Traditional herbal medicine and commercially available medicine are widely used. In South Africa, many patients still consult a traditional healer. And due to the natural origin of these traditional herbal medicines, many believe that they are safe, even safer than pharmaceutical products available. However, this is not true, as a herbal or medicinal plant is composed of various constituents as opposed to a pure active pharmaceutical. To this extent, we decided to determine the possible herbal hepatotoxicity of a crude aqueous extract from commercially available Zysmalobium undulatum, also known as Uzara, as one of the most popular traditional medicines in South Africa. We also decided we want to determine which system, HepG2C3As grown in traditional flat culture, or HepG2C3As grown as 3D spheroids, or an in vivo Sprague-Dawley rat model, will provide us with the most accurate and insightful information in regards to herbal hepatotoxicity. So we set up 2 experiments. In the first, we treated 2D cultures and 3D spheroid cultures for a period of 96 hours with daily dosages of Uzara and measured physiological parameters. Then we treated 3D spheroid cultures and the Sprague-Dawley rat model with Uzara for 21 days. The setup of each experiment is shown in detail in the following slides. For our first experiment, with the 2-dimensional cell cultures, we set up 5 experimental groups in 96 well plates. Cells were seeded at a density of 3,000 cells per well, and we measured physiological parameters, namely intracellular ATP, extracellular adenylate kinase levels, and protein content. The experimental groups were as follow: a crude Uzara extract at 200, 350, 500 and 750 milligrams per kilogram, and an untreated control group. The drug treatment was initiated 6 hours after seeding, and all experimental groups were treated for 96 hours. For the 3-dimensional experiment that we compared to the previous 2-dimensional experiment, HepG2C3H spheroids were prepared using agribels as previously discussed, and allowed to grow until 21 days to achieve metabolic equilibrium. We then set up 5 experimental groups in bioreactors, each containing exactly 239 21-day-old spheroids, to ensure that we have sufficient material for analysis. The experimental groups consisted of the following: Uzara aqueous extract at the selected concentrations, 200, 350, 500, and 750 milligrams per kilogram, as well as an untreated control. The physiological parameters, namely protein content, intracellular ATP, and extracellular AK, were measured in all experimental groups for a duration of 96 hours. For the comparison between the HepG2C3A 3D model and the in vivo Sprague-Dawley rat model, the experiments were set up as follows. For the 3D model, 4 experimental groups were prepared, with each group consisting of 4 biological replicates in individual bioreactors, and each bioreactor containing 207 spheroids. The experimental groups consisted of spheroids treated with Uzara at various concentrations, 200 and 250 milligrams per kilogram, spheroids treated with sodium valproate at 10 grams per kilogram to serve as a positive control for hepatotoxicity, and spheroids in normal growth medium as the untreated control group. The known hepatoxic valproic acid concentration was based on a value below the acute lethal threshold of 20 milligrams per milligram protein in spheroids as determined by Fay and Brzezinski. Physiological parameters, namely intracellular ATP, AK, and cellular growth, was evaluated at 24-hour intervals. Dosages were administered at 48-hour intervals for a duration of 21 days. The in vivo study was approved by the Animal Care Ethics Committee of the Northwest University prior to initiation of the study. If anyone wishes to see this ethical approval, you are more than welcome to email me afterwards. For the study, specific pathogen-free male and female Sprague-Dawley rats were obtained from the Northwest University vivarium, weighing between 150 and 250 grams and aged 7 to 8 weeks. The test substances tested on the Sprague-Dawley model were consisted of 200 and 250 milligrams per kilogram Uzara and 300 milligrams per kilogram valproic acid as positive control, and then a normal control group receiving only the vehicle. The test solutions were administered by means of oral gavage as 21 single dosages in 24-hour intervals. On day 7, 14, and 21, a sample population of rats, 3 male and 3 female, from each group were euthanized by means of decapitation, and terminal blood samples were collected for serum chemistry analysis. The results of these 2 experiments were very insightful. For the 2D versus 3D experiment, we first evaluated the protein content, as it provides us with growth as a function of time, and it also enables us to express all of our results in relation to the protein content. The figure on the left depicts the average protein content per experimental group for the 2D experiments. We observed growth inhibition for all the experimental groups treated with Uzara, when the protein content was compared to that of the untreated control group. The experimental groups treated with 200 and 500 milligram per kilogram extract showed more sustained growth, although slower proliferation appears to have occurred in comparison to the untreated control. The 350 and 750 milligram per kilogram extract treatment group showed a great measure of growth inhibition from time point 48 hours when compared to that of the untreated control group, as well as other experimental groups. At time point 72 hours, the 750 milligram per kilogram extract treatment induced significant growth inhibition when compared to other groups. The untreated control group data correlates well with previously published data suggesting that the doubling time of HepG2 C3a cells in exponential growth to be 3 days, and the optimal time for determining cellular effects is within this time frame. This also suggests that experimental conditions in 2D culture differ from day to day, and its usefulness is limited to the first 72 hours. The untreated control group growth characteristics correlates well with previously published data confirming the non-stable experimental conditions in 2D culture, especially for the first 72 hours. The protein content of the 3D spheroids are shown on the right. From the protein data, we notice that the spheroids experienced statistically significant growth inhibition at time points 48, 72, and 96 hours following treatment with Uzara at concentrations 350, 500, and 750 milligrams per kilogram when compared to the control group. The spheroids receiving 200 milligrams per kilogram extract showed moderate growth with a significant growth inhibition after 96 hours. On this slide, we can see the ATP data obtained from 2D cell cultures graphically depicted in the figure on the left, showing the average intracellular ATP levels in terms of average protein content, normalized to the untreated control group. Within the first 24-hour post-treatment, the intracellular ATP levels increased noticeably for 3 of the experimental groups treated with Uzara relative to the control group. Between time point 24 and 48 hours, all the experimental groups showed intracellular ATP levels similar to that of the untreated control group. However, at time point 48 hours, there was an increase in the amount of intracellular ATP for the 750 milligram per kilogram treatment group when compared to the other experimental groups. After 48 hours, the ATP levels increased for 3 of the experimental groups, 200, 350, and 750 milligrams per kilogram, with 750 milligrams per kilogram being significantly increased in relation to the untreated control between 72 and 96 hours. The 500 milligram per kilogram experimental group showed a slight increase in ATP at 52 hours, decreasing to values below the control at 60 hours, while reaching values similar to the untreated control group from time point 72 to 96 hours. The figure on the right gives a graphical depiction of the intracellular ATP levels per spheroid protein, normalized with reference to the control group. Within the first 4 hours post-treatment, intracellular ATP levels decreases noticeably for all experimental groups relative to the control group, steadily recovering back to the untreated control group levels over 24 hours. From 24 hours to 48 hours, there was a dose-dependent increase in ATP levels for groups treated with 200, 350, and 500 milligrams per kilogram extract, reaching values higher than those of the control group. However, the 750 milligrams per kilogram experimental group remained below the control, only reaching levels similar to the control at 48 hours. 52 hours post-treatment, the ATP levels again decreases in a dose-dependent manner. The experimental groups treated with Uzara extract at dosages of 200, 350, and 500 milligrams per kilogram resulted in a decrease in ATP with each successive dosage. ATP levels increased, however, to baseline levels or above within 12 hours. This decrease and subsequent increase of ATP levels noticed throughout the study may potentially be the result of an attempt by the cells to recover from the onslaught of treatment with Uzara. It is important to note that ATP levels for the 750 milligram per kilogram treatment group did not recover to the levels of the control group beyond 48 hours. This suggests impaired mitochondrial function brought on by the crude Uzara aqueous extract at 750 milligrams per kilogram. Adenylate kinase released from the 2D cell culture model is an indication of cell death, and in this figures we expressed it in terms of protein content. As you can see, the 2D cell cultures is depicted in the figure on the left. A slight decrease in AK released within the first 24 hours is observed for all experimental groups in comparison to the untreated control group. At time points 28 and 32 hours, all experimental groups showed an AK release similar to that of the untreated control group. At 48 hours, the groups treated with 350, 500, and 750 milligrams per kilogram showed a slight increase in AK release, returning to levels similar to that of the control group at time points 52 and 60 hours. Between 72 and 48 hours, there was an overall increase in AK release for all the experimental groups, with 350, 500, and 750 milligrams per kilogram reaching statistically significant increases at time point 96 hours when compared to the untreated control group. The figure on the right shows the AK release data for the 3D spheroids and indicates an initial increase in AK within the first 4 hours post-treatment for all experimental groups, and a subsequent decrease over the 24 hours when compared to the untreated control. At 28 hours post-treatment, AK again increased while steadily decreasing over the next 48 hours, and throughout the study to levels below the untreated control for all the experimental groups. However, 750 milligrams per kilogram extract treated resulted in a steady increase in AK release at 96 hours, indicating cytotoxicity. And this corresponds well to the diminished ATP levels and mitochondrial dysfunction seen in the previous slides. When we consider the interplay between intracellular ATP levels and active AK release, it appears that the cells grow under 2D conditions are initially able to cope effectively with the onslaught of Uzara at all the concentrations. However, towards the end of the study, the statistically significant increased AK and ATP levels indicates severe cytotoxicity. On the other hand, intracellular ATP levels and AK release from the spheroids indicate cytotoxic effects and the loss of cell viability as a result of treatment with Uzara on the spheroids for all experimental groups, but in particular the 750 milligrams per kilogram treatment. There is also an attempt of the spheroids treated with 200, 350, and 500 milligrams per kilogram extract to recover from each successive dosage administered. As we found the Uzara to be toxic during short-term use at high dosages, we attempted to conduct a subchronic study of 21 days on HepG2 C3A spheroids and the Sprague-Dawley rat model. The figure presented here indicated a significant concentration-dependent growth inhibition following treatment with Uzara at both 200 milligrams per kilogram and 250 milligrams per kilogram when compared to the untreated control group. The experimental group treated with 10 grams per kilogram valproic acid showed even more extensive growth inhibition. This growth inhibition corresponds well to the previous study with Uzara and other studies with valproic acid at various concentrations. The intracellular ATP levels expressed relative to the spheroid surface area are depicted in the figure on the left. There was a sharp increase in ATP levels for all experimental groups within the first 24 hours, but they decreased to levels below that of the untreated control group after 48 hours. ATP levels for the valproic acid positive control remained below the untreated control group, decreasing daily from day 5 to 14. By day 14, the intracellular ATP levels for the valproic acid group was significantly lower than that of the untreated control, as well as the Uzara experimental groups. Following treatment with the various concentrations Uzara, the intracellular ATP levels increased post-treatment, but decreased prior to each successive treatment. By day 19 and 21, ATP levels increased significantly when compared to the untreated control group. And this may be indicative of an attempt of the spheroids to recover from the continuous onslaught of treatment with crude Uzara. This corresponds well with the short-term study. The figure on the right shows the active AK release per spheroid surface area. An inverted trend can be observed between the AK released and the intracellular ATP levels. AK release appears to have decreased for all experimental groups relative to the untreated control within the first 24 hours post-treatment. AK increased to levels above the untreated control for the valproic acid, while remaining below the untreated control for Uzara groups. On day 6, there was a significant increase in AK for all the experimental groups, but by day 7, the AK levels for all the experimental groups decreased to levels lower than that of the untreated control again. AK levels remained below the untreated control group from day 7 to 14 for the valproic acid treatment group, but on days 15, 17, 19, and 21, the AK levels once again increased above that of the untreated control. The AK release for the Uzara treated groups remained lower than that of the untreated control for most part of the remaining treatment period. Again, we notice an interplay between intracellular ATP levels and AK released. The increase and subsequent decrease in intracellular ATP levels and the increase in AK release by the spheroids treated with the valproic acid is indicative of the toxic effect of this known hepatoxin and the loss in cell viability with continuous administration thereof. Results also indicated an initial loss of cell viability and increased toxicity for both concentrations of crude Uzara aqueous extract. In the Sprague-Dawley rat model, we found it ethically challenging to have daily measurements as we did in the spheroid model. We thus made use of biochemical markers used clinically in the diagnosis of liver injuries caused by hepatotoxins. Albumin, total bilirubin, and ALP levels for all the various experimental groups remained unchanged. Following treatment with Uzara extract at 250 mg per kilogram, AST levels increased relative to the untreated control group for the male participants on day 7 and on day 21 for the female participants, while ALT levels were increased at all time points compared to the untreated control group. The valproic acid group also had increased AST and ALT levels after 14 and 21 days treatment for both male and female test subjects. Treatment with Uzara at 250 mg per kilogram showed increased LDH levels for both male and female participants on day 7 and for male participants on day 21. The increased LDH levels were also seen for the male participants of the 200 mg per kilogram crude Uzara extract treatment group on day 21 when compared to the control group. The valproic acid treated test subjects also showed a cumulative increase in LDH levels from day 14 to 21 for both the male and female participants. Furthermore, an increase in triglyceride levels were observed at all time points following treatment with 250 mg per kilogram crude Uzara extract and also on day 14 for the individuals treated with 200 mg per kilogram crude Uzara. The valproic acid treatment group showed only a slight increase in triglyceride levels on day 14 and 21. So now we come to the conclusion. For both experiments, we made the following conclusion. In the first, the 2D versus the 3D, we could conclude that the Uzara extract showed an acute toxic effect at dosages between 500 and 750 milligrams per kilogram. Uzara at 350, 500, and 750 milligrams per kilogram also presented with significant anti-proliferating effects. When considering the behavior of the 3D and the 2D screening models, the results suggests that the 3D cell culture model may be a superior model in the elucidation of possible herbal toxicity compared to the conventional 2D cell culture model, due to the lack of the physiological relevance of the 2D model. Discrepancies in the data obtained from the 2D and the 3D models indicate that the 2D model may result in initial false positive and later false negative results, as the data suggests that the 2D model initially showed limited toxicity, possibly due to the different modes of DNA repair systems in the exponential growth phase. However, as the cells enter the decline phase of the growth curve, in the 2D cultures, they seem to be also severely affected by the Uzara at all concentrations, hence a false positive. In the 3D spheroid model, the majority of the cells within the spheroid structure are predominantly non-dividing, and only a few cells are believed to be in the G1 phase of the cell cycle. The latter correlates well to the in vivo situation and intact organs, where cells are also at various stages of the cell cycle or non-dividing, suggesting that a 3D spheroid model will give a more accurate representation of how an organ, in this instance the liver, will respond to a herb such as Uzara. The results of the study between the 3D model and the Sprague-Dawley to further evaluate the subchronic use of Uzara yielded the following. Both the in vitro 3D spheroid model and the Sprague-Dawley model was able to predict moderate hepatotoxic potential of Uzara. This results confirmed the usefulness of HepG2 C3a spheroid culture models to effectively and reliably predict potential hepatotoxic toxicity following long-term exposure to compounds. This model therefore offers a readily available in vitro model capable of bridging the gap between current two-dimensional in vitro screening models and the preclinical in vivo animal models. Implementing this screening model in the preclinical evaluation process of new drug entities could therefore reduce cost and time, as well as the use of animal models with compounds, use of animal models in the testing of compounds with potential hepatotoxic properties. We are not the only laboratories to have found discrepancies in terms of results obtained from 2D and 3D experiments. When evaluating anti-cancer compounds, Tai and colleagues found that the human pancreatic cancer cells grown as 3D microgravity spheroids were more resistant to apoptosis than that of their 2D counterparts, and exhibited LC50 values of known anti-cancer compounds closer to the levels observed in vivo. Breslin and O'Driscoll also found this to be true. Furthermore, a study by Lama and colleagues comparing the IC50 values of 41 known anti-cancer drugs for non-small lung cell cancer grown in 2D and in 3D spheroids showed that there were very low correlation, underlying the importance of culture technologies. And these are only a few of the available results indicating a need for a dimension shift in cell culture. You can decide for yourself now. Does dimension really matter? Thank you.
Thank you, Dr. Calitz, for your informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Question here. Why did you dose your two-dimensional and three-dimensional experimental groups in milligrams per kilogram?
That is a very interesting question, and thank you. I think the most common problem we have when we want to move over from in vitro studies to in vivo studies is that you cannot directly take the dosage and give it to an animal. It has to be extrapolated to an animal based on its mass. So what we decided to do is we decided to characterize our 3D spheroids and our 2D cultures in terms of protein content or spheroid size, and then we dose our experiments in terms of that, making it easier when we do go over to in vivo studies to just take that dosage, and then the results are more comparable because you're giving exactly the same dosage to the cells that you are giving to the animals. It's not like you're giving a large amount to cells, because if I give a large amount of compound to a cell, it will obviously be toxic. However, now I'm giving the exact amount of compound to the cells and the animals, and that is why we moved over to milligrams per kilogram. Thank you.
Thank you for explaining that. I would like to once again thank Dr. Calitz for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 27th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.